Hi guys, so today's video is all about receptors. How do you detect stimuli around you, such as light or pressure on your skin? We're going to learn about photoreceptors and the mechanoreceptor called Pacinian Corpuscle. So grab yourselves a notepad, pen and a cup of tea or your beverage of choice guys, and we're going to go right through it in a high level of detail. I'm going to finish the video with some exam practice so that you can see how this applies to those exam questions so you can smash your exam. Don't forget to like, comment and subscribe guys. Let's get into the video. Okay guys, let's get into this. So we're talking about A-level biology receptors today. So receptors detect a specific stimulus, and each type of receptor detects a single specific stimulus. So for example, photoreceptors detect light, chemoreceptors detect a specific chemical, and things of that nature. Now, examples of receptors include specialized cells called photoreceptors or proteins on cell surface membranes, like glucose receptors on cells in the pancreas. So a receptor can be a specialized cell, or it can just be a special protein with a specific tertiary structure complementary to something that needs to be detected. Now here we've got some protein receptors here on the phospholipid bilayer. So we can see we've got our nice phospholipids there. We've got this protein with a tertiary structure in purple there. And we can see the molecule that's being detected, for example, glucose, in red at the top there. So that will bind to the receptor and it will trigger responses in the cell. Now we're going to talk a little bit about receptors that specifically communicate through the nervous system now. Some receptors communicate with the nervous system, allowing the organism to respond to stimuli. And this is really important for survival. So organisms may need to see a predator to move away from it, or they may need to be able to see some food or something they need for their survival. So it's a really important response. Now, in the case of the organism moving away from the predator, the receptor will generate what we call a generator potential. And if you want more information on this, check out my videos on the nervous system, because I go through some really interesting stuff on the nervous system and how electrical impulses pass through neurons. Now, this generator potential can pass along the receptor and then it can stimulate neurons, which are the cells of the nervous system. Now, this can send messages throughout the organism, and the photoreceptors in your eyes right now are triggering electrical impulses going through your optical nerve to your brain as you watch this video. So how do receptor cells trigger electrical impulses then? Well, number one, when a nervous system receptor is at rest, it has a resting potential of minus 70 millivolts. And again, check out my videos on this for more information. I really break it down in a nice way. But for now, just know that when your neurons are at rest, they have a negative charge within them. And what then happens is when a stimulus is detected, protein channels open in the cell membranes of these neurons, and that allows positively charged ions to enter the cell. Now that's obviously going to make it more positive than the minus 70 millivolts we've got here. Now, we say that the cell membrane has been excited when these channels have been opened and we've got positive ions flowing in, and this leads to the generator potential in the receptor. Now, if the stimulus is large enough, it will lead to a generator potential that can trigger an action potential. Now, an action potential is when it goes from minus 70 to plus 40 millivolts. So the neuron becomes way more positive at that specific point. And that electrical impulse can then travel through the nervous system to communicate through the body of the organism. Now, stronger stimuli, so for example, really bright light as opposed to dim light, lead to more frequent action potentials. And that's why really bright light can give you more of an extreme reaction than really dim light. So we can see here that we've got the nervous system in a human, and we can see we've got the brain there at the top with the spine going down it. And that is our CNS, our central nervous system, because it's at the center of the body. Now, what we've got here on the right is a motor neuron. And essentially with the motor neuron, what will happen is we'll have positively charged ions entering the axon. And an example of these positively charged ions include sodium. And that's going to take it from minus 70, which we can see here in our action potential graph, right the way up to plus 40 if we get enough sodium, positively charged sodium ions, entering the neuron. So it's a really powerful tool. So next of all, we're going to cover the Pacinian Corpuscle because AQA really want you to know about this, and there's quite a few past paper questions that have come up on this topic. Now, the Pacinian Corpuscle is actually a pressure receptor in your skin. So if you press on your skin, you can feel pressure being applied to different parts of the skin. And that's because you've got these little pressure receptors, which are layers of tissue surrounding a neuron. So let's label it up then. We've got our sensory nerve ending here, running through the middle of the Pacinian Corpuscle. We've got layers of connective tissue called lamellae surrounding it. And then we've got the sensory neuron along the bottom. Now, number one, the Pacinian corpuscles detect pressure receptors in the skin. And because of that, we call them mechanoreceptors because they detect that mechanical force. Now, when we're thinking about spelling Pacinian corpuscle, I feel like Pacinian is fairly straightforward, but corpuscle is corpuscle. So corpuscle. Okay, that's the way I kind of sound it out to remember it. A little bit silly, but it helps me remember it. Now, number two, when the outer layers called lamellae, and we see the term lamellae in different structures in biology. So, for example, the fish gills have lamellae. Between the stacks of grana in the chloroplast, we have intergranal lamellae. So that is a term that comes up in many ways. And in this case, it just means layers of tissue. Now, when they become deformed, and by deformed, really, what we're talking about here is squashed, this opens stretch-mediated sodium ion channels in the sensory neuron cell membrane. So what we can do is we can actually squeeze in sodium ions into that sensory neuron. And you can think of it just like a sponge squeezing in that sodium ions, those sodium ions. And that's going to lead to a positive charge within the cells. And in fact, it's going to go from minus 70 millivolts, so don't forget the unit MV for millivolts, all the way up to plus 40 millivolts. Now, don't get too hung up on these numbers because different textbooks report it in different ways. So I've seen resting potential as minus 65, and I've seen the action potential as plus 30, plus 40, something around that region. But the idea is it goes from very negative to very positive. Now, number three, the sodium ions diffuse into the sensory neuron, and this is going to cause a generator potential. And if the threshold is reached, which is minus 55 millivolts, an action potential will be triggered and an electrical impulse will travel through the nervous system. And we can see here, we've got the sensory neuron, that's going to carry the electrical impulse to the nervous system, to the brain and spine. Now, what you need to know about next are that there's photoreceptors in the eye. And these photoreceptors, photo meaning light, receptor meaning to detect. So photo meaning light, think about photosynthesis, is creating with light. Photograph is a light picture. Receptors are going to detect light there. So we're breaking these words down. They're going to detect light. And you need to know about the cornea. You need to know about the iris, the pupil, the lens, the retina, the fovea, and the optic nerve. And also the fact that there's a blind spot where the optic nerve attaches the eye as well. So let's go through this in a little bit more detail. Now, I've numbered the diagram on the right, which will match the steps we go through on the left. So, number one, the cornea refracts the majority of light that enters the eye. So that's going to focus it in on the fovea over here, and we'll talk about the fovea a little bit more here. So we're going to be focusing light onto the fovea. So light is going to enter through the pupil and hit the fovea. Number two, we've got the iris, which is basically a muscle that controls the diameter of the pupil. And I always think of the iris as the kind of prettiest muscle in the body, because when you look at an iris, it's blue or brown or green even, and it's actually a muscle. Okay, it's got circular and radial muscles within it. Now, the pupil is simply a hole through which the light enters through. The lens, number four, fine focuses the light onto the retina, and the fovea is a really dense region of the retina that's full of photoreceptors. Number five, the retina is the layer containing the photoreceptors that detect light, and we've got rods that detect in black and white, and cones that provide colour vision. And I just think with cones, C for cone, C for colour vision, C for colour. So these are the two types of photoreceptors you need to know about, and I'll talk more about rods and cones in a moment. Now, the fovea has the highest concentration of cones in the retina. So that's really important for your crisp colour vision. Now, the optic nerve is the nerve that's going to carry electrical impulses from the eye to the brain so that you can actually process that information and it can give you vision. The blind spot next of all is where the optic nerve attaches. So we can see here we've got the blind spot there. The optic nerve attaches, so this is the optic nerve which will take electrical impulses to the brain. And there's no photoreceptors there because that's where the nerve attaches. So let's talk a little bit more about photoreceptors. Now, photoreceptors are transducers. Now, trans means to change or to move, okay? So a transducer is going to change light energy into electrical energy in this case. So a transducer changes something from one form into another. Now, photoreceptors convert light into an electrical impulse, which, as I've just said, makes them a transducer. And light enters the eye through the pupil and gets absorbed by the pigments in the rods and cones, which collectively we call photoreceptors. So right now you're using your rods and cones to watch this video and to do your revision. And well done for doing that, by the way. Now, this breaks down the light-sensitive pigments, which changes the membrane permeability to sodium ions. And because of this, it leads to a generator potential. So again, we have this influx of positively charged ions, which means we're going to go from our resting potential at -70 up to our action potential at +40. And that's going to allow for an action potential and a nervous impulse to travel through your nervous system so you can process that information. Now, if the threshold is reached, a nervous impulse travels along the bipolar neuron to the optic nerve. Now, we've got our rods here, and you can see the rods are rod-shaped, and we've got our cones, and we can see the cones are cone-shaped. They taper off into a point, and that's why they're called cones. Now, below them, we've got our bipolar neuron, and you can see that bipolar, it connects to two different neurons. So it connects both to the photoreceptor, which could be a rod or cone, but also to that ganglion cell. So when we think of bi means two. So if we think of, for example, a bicycle, that means two wheels, or binoculars have two lenses. Now, once it's gone through the bipolar neuron to the ganglion cell, it can then travel to the brain and allow us to see. So let's talk about rods versus cones, because in AQA A-level biology, this can come up. It really can. There's been loads of past paper questions on rods versus cones. So I've zoomed in a little bit here so you can see the rods and cones a little bit more clearly. And number one, rods and cones are both photoreceptors found in your retina. Now, I've produced a table here for us to look at them. So we can see that rods are black and white, whereas cones are colour. So remember, C for cone, C for colour. With rods, we've got one type, but with cones, we've got red, blue, and green cones. And each one is sensitive to a different wavelength of light. And that's what gives you the colour vision. Now, rods are more sensitive. And by that, what I mean is that it takes a smaller number of photons to stimulate a rod cell than it does a cone cell. So when it's dim, for example, dusk outside, everything looks a little bit grey. And that's because your rods are firing electrical impulses to your brain because there's just enough light to stimulate them, but there isn't enough light to stimulate your cones, so you don't have colour vision. Now, retinal convergence, it sounds posh, but it basically just means where we have more than one rod converging on the same bipolar neuron, okay? And if you want more information on this, check out my video on synapses. Because we can have 3 rods on the same bipolar neuron, we're more likely to reach threshold so that enough sodium can enter the bipolar neuron to stimulate an action potential and an electrical impulse. Now, with the cones, each cone forms a synapse with an individual bipolar neuron. Now, this will lead to less sensitivity, but it will give greater visual acuity, which means you have a higher resolution image produced by the cones. Now, the rods contain the pigment rhodopsin, which, as we said, gets broken down by light. So R for rods, R for rhodopsin, whereas the cones contain iodopsin. Now, visual acuity is a key definition you need to know, and that is the ability to tell apart objects that are close together. And retinal convergence means that more neurotransmitter is released at the synapse per photon of light. So it's more likely that the threshold of -55 millivolts will be reached at a given light intensity. So let's check out some exam practice next of all, because I always like to help upskill you with your exams. So question 1. The fovea of the eye of a hawk has a high density of cones per millimeter cubed. A hawk focuses the image of its prey onto the fovea. Explain how, so remember explain means to give reasons. So give reasons how the fovea enables a hawk to see its prey in detail. Do not refer to colour vision, so we do not want to be referring to colour vision in our answer here. So pause the video, have a go at this question, guys, in your notes, and then we will go through the answers. So the answers are, first of all, the fovea is rich in cone cells which have a high visual acuity. And we can see the term acuity is underlined there, which means it must be included for the mark. So the fovea is rich in cone cells, they have a high visual acuity. Each cone is connected to a single bipolar neuron. That will get you your second mark. You could say it's connected to a single bipolar neuron, you could say each cone is connected to a single neuron, or you could say each cone is connected to a single nerve cell. You would not get the mark for saying a single nerve because a nerve is a bundle of neurons. Now, next of all, cones send separate impulses to the brain through the optic nerve. If you get all 3 of those points, guys, you've mastered this exam question. Right, that's all we've got time for, guys. Don't forget to like, comment and subscribe, and I will see you in the next one.